The next question a lot of immunologists really want to learn is what other cells can be activated or how they can be activated also to kill the cancer cells. Overall, the biggest killer, the most important killer that is very important in cancer immunology is T cells, CD8 and CD4 T cells. They can do the killing. But there are other mm, cells that are also important. Here we mentioned myeloid cell, but you can imagine this also being cancer cells. If they present some antigens, this is kind of myeloid cells presenting the dendritic cells, also myeloid cell. They present the tumor antigen onto the surface, but this can be cancer cells directly. They also present antigens or mutations onto the cell surface. And these antigens, they can be recognized by T cells with T cell receptor, but they can also be recognized by B cells with antibodies, right? So once the antibodies bind to the tumor antigen, a number of things can happen. The first is that there's a process called the complement-dependent cytotoxicity. You can see here, once the antibody recognize the tumor antigen, they will recruit this CDC or the complement system, and then recruit another complex called MAC complex. And this MAC complex will again punch holes onto the cell surface and the cell will lyse and die. And so this is one way the antibody recruit complement and MAC to punch holes onto the target cell. The second approach is antibody-dependent cellular cytotoxicity, or ADCC. And so this is when the tumor antigen is being recognized by the T cells if it has the right constant regions. In this case, for example, IgG1 and IgG3, they will be able to interact with the Fc, the receptors, on the natural killer cells, so that they can bring the natural killer cells to the location. And the natural killer cells will kill the cancer cells. And the third is macrophage. This part is quite similar. When you have an antigen being recognized by the antibody, again from the Fc gamma receptor, it can recruit a macrophage. And when the macrophage comes, it doesn't directly kill, it engulfs, it eats up the target cell. And this is especially useful when you have a bacteria that is really foreign, and the macrophage will eat up the bacteria and then chew it up and spit it out and present those onto the cell surface. And so you can see B cells, they don't necessarily directly kill, but through CDC, ADCC, and ADCP, they can recruit other members there to do the killing. And so with natural killer cells, they kill with the following mechanism. So normally, all the normal cells have to express MHC class 1 to present antigen onto the cell surface. And T cell receptor makes sure that the presented antigen is not foreign. They have seen this before, and so they don't get activated if it's normal antigen. But natural killer cell also has a receptor to ensure that all the cells need to have MHC presentation, because one way for the cancer cell to evade T cells is to lose the MHC. And so basically, if the cancer cell does not present things onto the cell surface, then the T cell cannot recognize it anymore. And the body has a way to deal with this. Our natural killer cells ensure that all the cells have to present MHC class 1. If they don't, if they don't have ID, that's definitely bad. And so, if the bacteria come in, they don't display MHC class 1, NK cells can get activated very quickly to say, this thing completely do not have ID, let alone having the correct ID information. And so they do the killing. There are also, so you can see here, when the cell is missing the class 1 presentation, NK cell can kill. In addition, sometimes the cancer cell can have signals to when it's under stress. When the cell is under stress, there are ligands that are activating the NK cell. So the MHC class 1 is a suppressor. If this is present, the receptor can recognize this and NK cell does not get activated. And so if you don't have the suppressor, the NK cell will kill. Additionally, if the cancer cell is under stress, there are upregulated ligands, which will activate NK cell even more, then the NK cell will still do the killing. And so in this particular case, the cancer cell may not lose the MHC class 1, but if it presents an antigen and through ADCC can recruit the NK cell to the surface, the NK cell will be there and then see whether this cancer cell express some stress ligand on the cell surface. And if the stress ligand is activated, then the NK cell will again kill. Basically, the Fc gamma receptor already recognized this cell as expressing something foreign, and the cell is under stress, then the NK cell will know that this is a cancer or infected cell that presents foreign antigens, then you will kill it. And so a lot of these T cell activation and cancer cell relationship, you can also see that in the tumor infiltration and also the NK cell expression data from single cell RNA-seq analysis. And we mentioned another player, which is macrophage. Macrophage can engulf the cancer cell, but there are two different phenotypes of macrophage. In our blood that's circulating, we have very little macrophage. It's mostly called monocyte, which is a progenitor of macrophage. But once the monocyte infiltrates into normal tissues or into the tumor, it kind of differentiates into either an M1 macrophage or an M2 macrophage. And these two also have very, very different phenotypes. The M1 phenotype is more immunostimulation, inflammation, and anti-tumor activity. It's mostly to, it's similar response to recognizing a bacteria and trying to eat it up. And so the M1 macrophage is good. Once it recognizes the tumor, it will try to kill it. So it's more a similar response to a bacterial infection. Whereas the M2 macrophage is kind of more for tissue repair, remodeling, and it's more immune suppression. This actually will help the tumor growth. And this is a wound healing phenotype. And so normally when you have a cut, there will be some bacteria and germs coming in here. And there's like a lot of macrophage that goes there and kind of form like a cell band-aid. And so this allows the cell to kind of wound to heal, because as the wound is healing, there will be some things from outside. And if your immune system keeps on attacking and having too much inflammation, that wound will never heal. And so macrophage kind of, once the macrophage in the M2 is covering that region, it kind of insulates the outside from the inside, because outside there will always be some bacteria and bad things. With macrophage insulating there as a bandage, there's no point to get alarmed, whereas the inside will be kind of safe. And so the cancer cell have also learned this trick, and you can pretend to be a wound and force they secrete the cytokines to force the macrophage to differentiate from an M1 to an M2 type. And once the M2 type is in the tumor, the macrophage is no longer being activated. Those M2 macrophage are mostly suppressing the other immune system and not allowing the T cell to get activated. And so in each patient tumor, you can always see macrophage infiltration, some M1 and some M2. And very often we see that patient with more M1 macrophage phenotype usually have better survival and responding to immunotherapy. But if they have more M2 macrophage, it's usually pretty bad because they will suppress the T cells. And so you can see this is just one example of another study looking at single cell. You can do either single cell RNA-seq or CyTOF. They are looking at this is lung cancer, and they use specific antibodies even to select to kind of enrich for specific cell types. And then they do the single cell profiling. And then when they look at the changes in normal lung tissues versus lung cancer cells, and they can see the microenvironment really changing. For example, they see that there are clonal expanded PD-1 positive T cells, meaning that they can see the T cells becoming a little bit exhausted. They see an increase of CTLA4 high T regulatory T cells. Tregs is also bad. It kind of suppresses T cell activation. And they see the PPAR gamma high macrophage. This is kind of a macrophage go more towards the M2 phenotype. They see a decrease in NK cells, and they also see two different dendritic cell populations. So the CD141 dendritic cell is kind of a good dendritic cell. It's better at presentation to the T cells for T cells to recognize tumor antigen. But unfortunately, those T cells are decreasing, whereas the worst type of dendritic cells, CDC2 with high CD1C level, they are increasing. And so you can see the single cell analysis can really see these differences, suggesting that if there are drugs that can reverse this trend, increase NK, move the macrophage to M1, increase antigen presentation, and suppress Tregs, that can help with immunotherapy response.